This is DNA. It is made of phosphate groups, deoxyribose sugars, and nucleic acids bonded by hydrogen bonds. In this example, this is adenine, this is thymine, this is guanine, and this is cytosine.
To replicate this strand, first we have to break it apart using helicase. Helicase breaks the hydrogen bonds that hold the two strands together, leaving us with the two original strands that we will synthesize DNA off of. However, since DNA is so tightly wound, it wants to close or tangle in on itself. The depoisomerase holds the DNA so it doesn't unwind or tangle. This triangle formed is called the replication fork. Focusing on the leading strand, we can see the RNA primer. The primer is a short strand of DNA that is needed so the nucleotides have a place to start synthesizing from. Then the DNA polymerase attaches and starts to attach nucleotides. This strand is called the leading strand because it leads with faster synthesization. This occurs because DNA can only be added in the 5' to 3' direction, which the leading strand is following. The lagging strand is a bit different from the leading strand. This strand has to synthesize from the 3' to 5' direction, which isn't possible, so the lagging strand synthesizes in fragments called Okazaki fragments. An RNA primer attaches to the original strand of DNA to allow the DNA polymerase to synthesize the lagging strand. This is followed by the ligase, which joins the Okazaki fragments together to form a complementary strand. As the strands form, we can see why DNA replication is called semi-conservative. As you can see, the newly synthesized DNA has one strand from the original sequence, and the complementary strand is the new DNA strand. Since it only conserves one part of the DNA, it is called semi-conservative. If this process were to be RNA transcription, the only thing that would change is the DNA polymerase would be RNA polymerase. Rather than attaching DNA nucleotides, it attaches RNA nucleotides. Adenine, guanine, cytosine, and uracil, making a strand of RNA rather than DNA.